Yes, okay, thank you. Thank you. Okay, so
Okay.
please,
Um,
floor is yours.
okay. Yeah. Thanks, Janusz. Thanks for inviting me. And I know some of you must have heard this talk already last month when I gave this in Morn. Maybe you were there in person or you saw a recording. But I'm going to present things slightly differently. It's the same topic. I'm going to cover the same ground, but I'm going to uh, try to present things somewhat differently. Maybe it helps catch some points that you missed last time. And of course, if you're hearing this for the first time, then that's great. Just ask me at any point if you have questions. So do I understand correctly, Janusz, that I have maybe 45 minutes or so to talk
Yeah,
about the main?
yes, that's it.
Okay, okay, great. Um, Okay, so yeah, I'm going to talk about bifunctional enzyme action as a source of robustness, and this is going to present a new kind of way of thinking about reaction networks. Okay, so this is joint work with Tung. I think he's in the audience.
Yes, he is
And
present. Yes.
okay, so right, so kind of to set up the problem, let me give you kind of the biological background. So um, this kind of so we are interested in robustness, which is something that is important for the functioning of biological systems, and in particular, if we think about signaling systems, um, there is you should think of this here as the surface of the cell. There is a signal that is coming in from outside the cell, and that is being transducted to the inside of the cell. So a signal moves from outside to the inside, and then the cell produces an appropriate response, right? So this is how cells communicate. And there are these signaling proteins that are on the surface of the cell. A ligand comes and binds to the signaling protein, and then that produces some kind of a response inside. Okay, so there are many different kinds of proteins that are involved in signal transduction, and here is a list of some of those proteins, their names, the number of molecules in each cell, and the standard error. And what is particularly striking in this table here is that if you look at the error, that's in some cases as high as 20%, right? So the number of even between two cells of the same type, variation can be quite large. So the number of these signaling proteins can vary up to 20%. Now, what that means is that the amount of signal that is transducted inside the cell can vary by 20% or more, right? And so if I just take any ordinary signaling circuit, the response that will be produced will vary from cell to cell, right? As the protein numbers vary, the response also varies. Now, this is not a desirable feature. Ideally, we want the response inside the cell to depend on the strength of the signal, but not on what is going on inside the cell. What are the sort of the noise part, the number of proteins, or any kind of environmental fluctuations? We don't want that the response depends on that. We just want that the response depends on the signal. So that clearly requires some special design to to so that to ensure robustness against these types of fluctuations in protein numbers. So if I have some kind of a special design which produces robustness, then all cells will have the same response curve where the output is proportional to the input, but there aren't cell to cell variations. Okay, so to kind of put this in mathematical terms, it's useful to look at an example. So here I have one example of a reaction network. Um, biochemical reaction network. And here, so I have this reaction IP plus E goes to IPE, right? They form this complex. There are these arrows which indicate reactions, and then the labels on these arrows, they are reaction rate constants for something called mass action kinetics. So most likely everyone has seen mass action kinetics, but it's just it's a rule that tells you how fast the reaction happens. So once I have such a rule, I can write down differential equations, ordinary differential equations, which govern the dynamics of the concentrations of these species. So these are square brackets are all concentrations, species concentrations. For example, this is concentration of the species IP, and these are differential equations. Okay, so once we have differential equations, we can observe a few things. One is that there are two conserved quantities. The total substrate um, is conserved. So even though substrate changes forms, and I'll come back to this, I'll tell you what I mean by a substrate and what I mean by an enzyme. But basically, enzyme is, in short, enzyme is something that helps the reaction happen. So here I have an enzyme E, which helps this reaction go forward, and the substrate is what is changing in a reaction. So the total amount of substrate is conserved and the total amount of enzyme is conserved. So even though substrate may change form, the total substrate is always the same. It's independent of time, and the same with enzyme. The other thing you can do is solve for a positive steady state, and you find that the concentration of the species I is this function of rate constants, right? So in particular, this concentration of I at a steady state does not depend on the total substrate or the total enzyme, right? So as even though these concentrations can vary, and that's kind of novel or surprising, right? Because here I appears in this conservation law for total substrate, but somehow its its concentration at steady state remains invariant while the others are changing. Okay, so this property that the concentration of a certain species at steady state is invariant across steady states and does not depend on the total substrate or total enzyme, that is called absolute concentration robustness. And I will abbreviate that as ACR. This is because it's a common abbreviation. Okay, so this uh, property was defined in a paper by Shinar and Feinberg in 2010, which appeared in Science and has been cited by many publications. When I last checked, maybe 1 month ago, it had already 473 citations. And they did, I would say, three things in this paper. One was introducing this term, absolute concentration robustness. Even though this had been studied before, they introduced this phrase, which has become popular. They defined ACR as a species which has the same concentration in every positive steady state. And they gave a theorem which gives easily checked conditions for establishing that a network has ACR. Okay, so there are basically uh, two main conditions and one, I would say, auxiliary condition. So the conditions are that the network has deficiency one. I'm not going to define deficiency because this is not really, I mean, this is somewhat tangential to my talk, but this is a condition that can be easily checked for any given network that it has deficiency one. And two non-terminal complexes differ in one species X. Again, this is very easily checked. Once you have a network and you read the definition, you can sort of check it in a matter of a few seconds. Um, and if you also know that a positive steady state exists, then immediately you know that the network has ACR in that species X. Okay. So So this broadly I'll call the deficiency-based approach, and Shinar and Feinberg, they clearly articulated ACR, and they gave these conditions which are precisely defined and easy to implement. Now notice that I said that this works, this theorem, one of the conditions is that you need deficiency equal to 1. Deficiency is a non-negative integer and it can take any values, 0, 1, 2, and so on. But the theorem is only restricted to deficiency 1. Um, because this theorem is so sort of simple to state and simple to check, I would say that many attempts have been made to look at what happens uh, for higher deficiencies, and those attempts have generally not been successful. So there is no sort of extension of this theorem to higher deficiencies. And I think there is good reason for that, which is that deficiency itself has no biochemical interpretation. I often find that, at conferences, maybe some graduate student who's just starting out or undergraduate, they complain that they don't understand deficiency. And partly it might be that they don't understand the mathematical definition, but partly it's also that um, it's does not have any kind of clear interpretation in terms of chemistry or biochemistry. It's a mathematical property. Okay. And one more thing I would say is that the existence of steady state to be able to apply Shinar-Feinberg conditions, that must be inferred separately, right? So you need other considerations to establish existence of a steady state. All right, so a couple of years ago, Tung and I, we decided to look at a certain class of networks called covariant modification networks and just establish which of these networks have ACR to try to find conditions that are sufficient for ACR. So we started with this so-called futile cycle, where some substrate changes from S1 to S2 all the way to Sn, and then there is another backward chain where you go from Sn to Sn minus 1 all the way back to S1, right? So these are different forms of a substrate. And in the forward chain, they all chain, the enzyme is E, and then C alpha is another enzyme, which is one of the complexes appearing in the forward chain. And we found conditions for ACR in this type of a network. The thing I want to kind of focus on right now is that the futile cycle has deficiency equal to the number of steps in either one of these, either the forward or the backward chain. So I have n minus steps in the forward chain, S1 to S2 to S3 all the way to Sn, and that is equal to its deficiency. So basically, deficiency can be any number for this futile cycle. You can also look at variations of this futile cycle. So you may have something like this, or maybe where dephosphorylation happens in one step. Uh, Dephosphorylation sort of goes through a different path than phosphorylation. And you can, extend this. You can extend the number of steps. And again, the deficiency can be anything. And so we did not see any link between deficiency and um, the presence or absence of ACR. Okay, so what this suggests is that deficiency is just unrelated to ACR. It may be helpful for proving some theorem, but it's otherwise it has no connection. And so we need a new approach. Um, so it turns out that the new approach, in some sense, there is already a foundation for that new approach, which is um, something called bifunctional enzyme action. And this has been well studied in literature that combines experimentation with mathematical modeling. So this is just this is not a complete list of papers, but many of these papers, what you will notice is in the titles there is a phrase. For example, in paper one, there's opposed reactions. Then there is bifunctionality in number five, and then there is paradoxical components. These are all um, ways of talking about something called bifunctional enzyme action. And it turns out that this bifunctional enzyme action is actually very important for ACR, as I'm going to talk about. So this is kind of a starting point. Okay. So um, bifunctional enzyme is basically an enzyme that has two and often opposite functions in a network. So a regular enzyme, it's a regular enzyme in one location in a network, uh, while at another location, the enzyme it acts as an enzyme after forming a complex. So the enzyme and the substrate they bind together into a complex, and then that complex itself acts as an enzyme, right? So an example of that is this reaction network here, where some species IP meets this enzyme E, forms a complex IPE, and then that complex IPE acts as an enzyme that phosphorylates I and sort of pushes in the opposite direction, right? So makes I turn into IP. So this is a bifunctional enzyme. E is a bifunctional enzyme because in one form, it helps IP become I, and in another form, it helps I become IP. What I mean, intuitively, what this does is the bifunctional enzyme, it kind of produces control via this balanced push and pull, right? So it pushes and pulls so that you somehow get kind of a balance. Um, okay, so if we think about this bifunctional enzyme-based approach, then deficiency is not considered at all. This, as far as I can tell, this exists as isolated applications. There's no sort of general theorem or algorithm that can just be applied to any network. So, what we would like to do is what I'm going to talk about in this talk is we want to have a precise theorem that connects with bifunctionality mechanism and something that works for all biochemical networks. Okay, so what is a biochemical network? What do I mean by biochemistry? Now, biochemistry is sort of conventionally classified into these three branches. One branch is structural, while the other two branches are more related to enzymology and metabolism. And they have a very special character. So if you think about the reactions that appear in biochemistry, the species, they all have some type, right? So there are species that are substrates, which are stars of the show, right? I mean, they are the ones who kind of you observe or you are interested in, right? They change from one substrate to another. Enzymes, which are directors, which kind of basically lead the action. And then substrate and enzymes, they form compounds, right? So these are kind of the supporting cast. The reactions are also have types. So the most common type is substrate transformation, where one substrate S turns into another substrate S star. A very common mechanism for this is the Henry-Michaelis-Menten mechanism. I'm sure people have seen this, where a substrate and enzyme they combine reversibly to form a complex C, and then that complex can disassociate and go back to the original substrate or produce a new substrate S star. This is called the Michaelis-Menten mechanism or the Henry-Michaelis-Menten mechanism. There is the transfer reaction where a phosphate is transferred or some other group, phosphate or something else is transferred from a substrate X to another substrate Y. Again, I'm just showing one particular mechanism for a transfer reaction. Here I'm showing without an enzyme, but you might have an enzyme here as well. Okay. Um, and here is a bigger list, a complete list of reactions which have at most two substrates on one side, right? So you have a transformation, transfer. You can also have cleaving, binding, inflow, outflow, sequestration, pair production. And this is a complete list if I only allow at most two substrates on one side. If you allow more substrates, which is a less common situation, of course, the list is much bigger, right? And to give you an example of how common this is, um, the sort of the most, one of the most well-studied reactions is glycolysis. And if you look at the overall reaction, that's a cleaving reaction where a glucose splits into two molecules of pyruvate. So I should mention here that when we talk about cleaving reaction, Y and Y star need not be distinct. So I might have X going to 2Y, as is the case for glycolysis. Um, Now, if you zoom into glycolysis and you look at the standard textbook presentation, it's usually broken down into 10 steps, right? And each step has many reactions in it. So, for example, transformation reaction, it appears in steps 2, 5, and 8. Transfer reaction appears in steps 1, 3, 6, 7, and 10, and so on, right? So all of these reactions have one of these types, and all the species fall into one of the types that I showed you in the previous slide. So again, biochemical reactions have a very special character, um, and that's worth keeping in mind. Okay, so I said S to S star is a reaction, it's a transformation reaction, and then this here is the Henry Michaelis-Menten mechanism. And so I want to kind of clarify this language. Um, when I say reaction, I mean a composite reaction where the substrate, I'm talking about what happens to the substrate. The mechanism for that reaction, there can be many different mechanisms. If you look at a composite reaction, in fact, from paper to paper, you'll find people using different uh, mechanisms for the same reaction. And there is good reason for that. You can establish the overall reaction much more easily and with much higher confidence than you can establish the mechanism, right? So to say that I have this HMM mechanism or a mechanism with two or three intermediates, that's a much more difficult task for an experimentalist and a modeler, right? So I might have something like two intermediates where S plus E form a complex C1, then that turns into another complex C2, and then you get S star plus E. Or I might have something complicated going on in the middle. And these are just three mechanisms that I found in some paper. Um, you might even have that the substrate and enzyme, they always form a complex reversibly. For example, here S plus E, they form C, S star plus E, they form C prime. And again, this is always reversible, but still the overall reaction is irreversible because C to C prime is irreversible. You might even have that the intermediary compound or this compound is not really an intermediary. It kind of appears in a side branch, but it still may play some role in the overall model, right? So I'm just giving you some examples of detailed models, but for all of these detailed models, we have the same reaction, composite reaction, right? So again, in terms of vocabulary, we have a composite reaction, which is a high confidence model. The reason this is high confidence is because to establish a composite reaction, all I need to do is put a substrate and an enzyme together. And if all the substrate turns into this S star in the presence of the enzyme, then I know what the substrate is, I know what the product is, and I know what the enzyme is. If in absence of the enzyme, this reaction does not take place, then again, that tells me that that's the right enzyme. But to know what the detailed model is, I need to look carefully at the product formation curve. I need to do careful measurements and modeling. So the detailed model is always going to be low confidence. And the detailed model is made up of a number of what are called reaction steps, right? So these are individual steps in a detailed model. These are reaction steps instead of a reaction. Okay. So what is an appropriate detailed model? I won't write down the precise definition. You can find it in our paper once it comes out. But basically, um, a detailed model, it breaks up into a reactant block and a product block. And the product block, which is this red box, is a connected component, while for the reactant block, it's either a connected component or I have a sequence of arrows that goes from S plus E into the product block, right? So almost every kind of detailed model that you find in the literature is going to satisfy these conditions, right? I mean, they're fairly general conditions. One example of a model which is not a detailed model for the transformation reaction S to S star is this, what I'm showing at the bottom of the slide, the so-called reversible Michaelis-Menten. And the reason is that this does not decompose into a reactant block and a product block, right? What I need is one arrow going from the reactant block to the product block. And this, at the bottom, this reversible Michaelis-Menten does not have that property. And in fact, if you think about this reaction, this is really different because if you start with a certain amount of S, that is not going to get all used up and convert into S star. In fact, you will reach some sort of a steady state where you have a positive concentration of S and positive concentration of S star. So in fact, you cannot even say that S is the reactant and S star is the product, right? They're completely symmetric. So this is not a detailed model, okay? But everything else is. All right. So because I'm interested in knowing what is happening to the substrates, I'm going to describe that using this hypergraph framework. I'm going to describe the substrate skeleton. So if I have this reaction, S plus S prime goes to P plus P prime, that reaction is a hyper edge that connects the set S comma S prime to the set P comma P prime, right? So a hyper edge is a generalization of an edge which connects, generalization of a directed edge, which connects a non-empty set to another non-empty set. Instead of just connecting one node to another node, it connects a set of nodes, SS prime, to another set of nodes, PP prime. And I'm going to show a hyper edge as this kind of pair of split edges, but this is only for visual depiction, right? I mean, you should think of this as basically the set SS prime going to the set PP prime, right? The split edges is sort of convenient because we want to be able to draw a graph just like we draw an ordinary graph, but there's nothing kind of special about this arrow S to S prime to P prime. There's just one hyper edge that goes from SS prime to PP prime. Okay, similarly, I have an hyper edge for the reaction S plus S prime going to 0, S going to P plus P prime. This is a cleaving reaction, binding reaction, and so on. Okay, so if we look at this IDH-KP IDH signaling system, which is one of the reaction networks that appears in the Shinar-Feinberg paper, you can make the substrate skeleton for this, right? So in the top reaction, I have IP, the substrate IP being converted to I, right? So I remove all the details of enzymes and intermediate complexes and everything. And so the overall reaction is simply IP turning into I. The bottom reaction is I turning into IP. So the substrate skeleton is just this, IP to I to IP, right? That's the simplest reversible skeleton that you can have, or simplest weakly reversible skeleton that you can have. Another model, NZ-OMPAR, there are two detailed mechanisms of that presented in the Shinar-Feinberg paper. And if you make a substrate skeleton for either of these mechanisms, it's actually exactly the same. So there is one reaction where X becomes XP, right? Whether you look on the left or the right. There's another reaction where XP and Y combine to give you X plus XP. So that's a transfer reaction. So there's hyperedge 1, hyperedge 2, and hyperedge 3, where YP turns into Y. Here is the skeleton for an n-step futile cycle, right? So it's just substrate S0 turning into S1 to S2 all the way to Sn, and then the backward cycle where Sn goes to S0. I might have that the dephosphorylation occurs in one jump, so the skeleton would look like this. I might have an additional step in the backward chain. So I go from S3 to S2, so an extra step, S2 to S1 and S0. I might even have some kind of a complicated thing where I go from S0 to S1 to S2 to S3, but then when I come back, I go from S3 to S1, S2 to S0. So all of these are just examples of skeletons, hypergraph skeletons of different types of these futile cycles and its variations. Okay, so to state the main results, I need to define a couple of things. The first one is something called a current on a directed hypergraph, right? So current is a non-negative quantity that is assigned to the hyperedges such that the current in is equal to current out at every node. Now, remember, a node is just a substrate, right? So it's always one substrate, every node. And so to define current, I only need to look at the graphical properties. I don't really need the identity of the nodes, right? So it's a non-negative quantity that is assigned to the hyperedges such that current in is equal to current out. And if the current assigned is positive on every hyperedge, then I will call that a positive current. Okay, so I'm going to say that two hyperedges are current balanced if a positive current assignment exists, and if for every such positive current assignment, the two hyperedges have equal current. So some examples of this, this cycle, a single cycle, right? I mean, the simplest reversible skeleton, that the two hyperedges are current balanced clearly because at this node, I must have current in equal to current out. So the two hyperedges must have the same current. Here I have what looks like two cycles kind of sitting on top of each other, although there are just three hyperedges, right? 1, 2, and 3. Again, because of node balance at this top left node, I have that the first two hyperedges are current balanced. And because of node balance at the bottom left node, I have the hyperedge 2 and hyperedge 3 are current balanced. So all three hyperedges are current balanced. Same thing here in example 3. Because of current balance at nodes, all three hyperedges are current balanced. Okay, the last example is a bit complicated. And in order to show clearly what the hyperedges are, I used the same color for split edges that are basically the same hyperedge. So I've used the same color red for this hyperedge. These two arrows, they are the same hyperedge. So we have 7 or maybe 8 hyper edges, 8 reactions, and you can write down the uh, balancing conditions at each node. And what you find is that this I1 and I2 are current balanced. That's sort of easy to see because of node balance at XP. But you also find that these two edges, let's see, I4 and I7, which are not next to each other, right? I4 is this blue edge, I7 is this black edge. They are kind of far away in the network, but they are also current balanced. And you can get that from solving these node conditions. Okay. Um, the other thing I need is a bifunctional pair. So I already talked about bifunctionality earlier. A bifunctional pair on a hypergraph is just a pair of reactions or hyperedges where a bifunctional enzyme is acting, right? So if I have these two reactions, um, in the first reaction, this enzyme E is acting as a normal enzyme, and it forms this complex IPE, which then becomes the enzyme for this reaction I going to IP. So this pair of hyperedges is a bifunctional pair, and the first edge, I'm going to call that an agonist. So IP going to I is the agonist, while I going to IP, that's the antagonist, right? They sort of push and pull these two edges. And so that's why they are the agonist and antagonist. Okay, so in order to establish what is a bifunctional pair, I only need the identity of the bifunctional enzyme, nothing else. So I need the skeleton and I need the identity of the bifunctional enzyme. I don't need to know the detailed model. That is important to note here, right? So even though I used a detailed model as an example, I just need to know that this is a bifunctional pair. I don't really need to know the details. Here, the detailed model could actually be something else or could be much more complicated. All right, so now we are ready to state the main results based on this hyperedge or substrate skeleton formalism. Okay, so the first is we need existence of a positive steady state to talk about ACR, and that comes out of this formalism. We don't need separate considerations for that. So if there is a positive current on the skeleton, then the mass action system of any detailed model, again, I don't need to know the detailed model, right? The mass action system of any detailed model has a positive steady state for some rate constants, right? And so this is, again, this is very useful because the detailed model may actually not be agreed upon. There may not be a consensus view, or it may not be known, or may even be varying from one place to another. So mass action system of any detailed model has a positive steady state for some uh, rate constants. Okay. Before I state the next result, I want to consider a slight generalization of
Excuse me,
ACR.
excuse me,
Yes,
brother.
yes.
How do you check the sufficient condition in the previous statement? The existence of a positive current.
Right. So basically, you kind of just work with this hypergraph. So for example, all of these, uh, for all of these examples, I know that a positive current exists. So essentially, I just, I mean, for example, here, right? I, I just assign the currents, and these two currents must be equal. So and they must be positive.
Mhm.
Um, I, I mean, I guess it's a computational problem on this graph, right? And it's a I think it's I don't know if I have any comments on kind of the complexity of this problem, but But essentially, yeah, I mean, you just check it from the node balance. So it's a kind of a graph problem.
Okay, thank you.
So yeah, okay. Um, okay, so to talk about robustness, I need this uh, generalization of ACR, right, which is concentration robustness of a complex. Okay, so I'm going to say that a linear combination where the coefficients they come from non-negative integers. So essentially, in reaction network theory, this is called a complex. A linear combination summation AI xi is robust if this quantity, uh, the concentration of xi to the power ai is invariant across all positive steady states. And so if the complex is just a single species, then this boils down to ACR. Okay, so suppose I have that a pair of hyperedges in the skeleton satisfies two conditions. One is that it is current balanced, the pair is current balanced, and it is a bifunctional pair, right? It's this agonist-antagonist pair. Then the tail of the antagonist edge has concentration robustness in the mass action system of any detailed model. Again, you don't need to know the detailed model. Uh,
Mhm.
so for example, here I know that for the first example, this pair of hyperedges is current balanced. And if it is also bifunctional, then the tail of the antagonist will have current balance. Again, example two is all the edges are current balanced, right? So example one and example two are in fact the two main applications in Shinar and Feinberg. And they also turn out to be sort of the two simplest examples of skeletons that you can write down. So one is this IDH KP IDH system, and the second is MZ on par. The third example is a variation of a futile cycle. Again, all edges are current balanced. And the last example is where I have two current balanced pair of edges, I1 and I2, and I4 and I7. So again, if either of these, for example, if I4 and I7 is a bifunctional pair, and if I4 is the agonist and I7 is the antagonist, then this system will have ACR in YPP, right? Or concentration robustness in YPP. But since YPP is a single species, you'll have ACR in YPP. Okay, so let's look at a couple of somewhat more complicated examples. So I have three transfer reactions now. And again, the same color means it's the same hyper edge. So you should imagine this network on a Mobius strip because I have one hyper edge where x plus yp goes to xp plus y, another hyper edge y plus zp going to yp plus z, and then you twist this xp so that xp plus z goes to x plus zp, right? So I have three hyper edges, and there is a cyclic phosphate transfer going on here. So X to Z, Z to Y, Y to X. And these are the three composite reactions. And it's easy to check that all three hyper edges are current balanced. And so if there is a bifunctional pair in there, then the tail of the antagonist, which is going to be a, a complex that's made up of something like x plus yp, that will have concentration robustness. Okay. And the last example is where so this all this formalism applies even when you have stoichiometry that's higher than one. So for example, if I have a composite reaction 2YP going to YPP plus Y. This is an example of a transfer reaction where a phosphate is transferred from one of the YPs to the other one. You just imagine a hyper edge from a multi-set, not a regular set, but a multi-set YP comma YP to another multi-set YPP comma Y, right? Now, to draw this as a graph, I use this kind of a dot, right? So there are two edges coming out of YP. They sort of meet in a dot. This basically denotes a multi-set, and then they split into YPP to Y, right? But again, this hyperedge formalism extends to stoichiometry greater than 2. And so a hyperedge is something that you should imagine connecting one multiset, one non-empty multiset to another non-empty multiset, right? And again, it's easy to check for this example that all hyperedges are current balanced. And so if there is a bifunctional pair in there, then the tail of the antagonist will have concentration robustness. So for example, in this case, if it happens that this purple edge is the antagonist of a bifunctional pair, then you will have concentration robustness in 2YP, which is actually absolute concentration robustness in YP. So even though the complex is not a single substrate uh, complex, it's still you still get ACR in YP. Okay, so just a few final remarks. This formalism applies to any biochemical network, right? I mean, and so biochemical network here, biochemical network is sort of the, as I described, it has a certain type or a character, right? There are certain types of species, types of reactions. And so this formalism is going to apply to any such network. Bifunctional enzyme action is the key biochemical mechanism, right? So this is something that biochemists would understand as a mechanism, and we've basically connected it to a theorem, right? And the way we could connect the mechanism to a theorem was by defining something called a current on a directed hypergraph, right? And what the current gives you, what it does is it connects distant edges or possibly distant edges in the skeleton with each other. So they get connected via the current. Okay, and the detailed models that we consider can and do have any deficiency. You are not restricted to deficiency 1 or 0 or the load deficiency cases. All right, so uh, lastly, I should mention that you do see other kinetics besides mass action kinetics in literature, and all of this can be generalized to non-mass action. The only thing we need for this formalism is that you have linearity in enzymes and in the enzyme substrate compound. But you don't need linearity in the substrate itself. For example, Michaelis-Menten kinetics is totally fine because it is of this form, which is saturating in S. It does not grow monotonically, does not grow in S, it saturates in S, but it is still linear in E and in the enzyme. And so that's totally fine. All of what I said earlier holds for Michaelis-Menten and Hill kinetics. Okay, so to wrap up, I showed one example uh, of a futile cycle that is already published in a paper published last year, 2024. Most of the work that I presented today is completed. We are yet to submit it, but we are in the process of doing that. And so um, you should be able to see it in a few weeks online. And so, yep, I would like to end by thanking NSF and thanks to all of you for listening.